better to represent this than Professor John Camm. He has so many titles and honors that I can't even pronounce some of them because my British English isn't good enough. But I first became aware of John Camm as someone who was a specialist in arrhythmias. And, you know, there's a great deal of excitement now about the wearables and the little iWatch and stuff like that. And actually, I think I saw the first device in Cape Town at a dinner in Camps Bay when you had an iPhone, an early iPhone, primitive one, that had a little EKG monitor. So you've always been on the leading edge, near the limits. So it's perfect that you're going to be speaking about drugs and devices. What are the limits to cardiac dysrhythmias?
Thank you very much, Peter and Mark. Good afternoon, everyone. I'm very honored to have been asked to deliver this lecture, and I thank Derek and the organizing committee for giving me this opportunity. As you see, I have a tough task, the whole of cardiac arrhythmias in a few minutes. So I've had to choose just a few topics that interest me particularly because they are limiting issues, and we'll see how we go from there. Firstly, of course, I have to show you the conventional conflict of interest slide. Now, this slide gets longer and longer as people expect you to write most of your biography upon it. But of course, all that serves is to obfuscate the bit that you're really interested in, which is the lower part, which are companies with which I've been affiliated in the last 5 years or so, and where money has changed hands either for grants or for advice. Please take a note of these companies and remember that, if you can, as I give this discussion. Firstly, let's think about anti-arrhythmic drugs. The story goes back to 1914, when quinidine came on the scene. And it stopped, really, in about 2010, when vernicalent was released onto European markets, not into North America. You can see that there's some hesitation in this development scheme, and that hesitation was occasioned by the classic trial known as CAST, the Cardiac Arrhythmia Suppression Trial. It was a trial which was essentially testing a hypothesis of whether ventricular premature beat suppression would improve the likelihood of survival post-MI. But it had contrary results, as we'll see in a moment, and that meant that a large number of drugs that were then in development were suddenly stopped from their development, except for a few, which somehow slipped through the net. And we have cybenzoline and pelsicanide, which are available in France and in Japan, and tazolene in Poland, and ethmazine in Russia. But we are not seeing these drugs here or in North America. You can see that after the CAST study, most of drug development suddenly became oriented towards atrial fibrillation. And that therefore led to new challenges in drug development. And we're waiting now to see how some of these challenges may work out. But let's first recall the CAST study. You'll remember that ventricular premature beats occurring after a myocardial infarction were always thought of as potential triggers for sudden cardiac death. And the hypothesis was that if these could be suppressed, and we knew already that drugs like flecainide and encainide would suppress these ventricular premature beats, would they also manage to reduce sudden cardiac death in the patients? That proved not to be a true hypothesis, and the opposite was true. It was the first time that proarrhythmia, which had first been mooted by Seltzer and Ray in 1964 when they described torsades de pointe with quinidine, had shown a remarkable clinical adversity. We also found that, of course, ischemia seemed to be more of the trigger for these flecainide and incanide-induced cardiac arrhythmias. If we look into PubMed to see what happened to drug development with antiarrhythmics, we can see that by the time 1989 occurred, the development had peaked and publications began to fall off. Of course, this fall off was largely confined to ventricular tachycardia and ventricular fibrillation, etc. And drug development for atrial fibrillation, however, continued. If we look now at what kind of clinical trial we were used to seeing, it was like this. This is a particular good example because it's a placebo-controlled trial, also with an active comparator. We're looking at 2 antiarrhythmics and a placebo given to patients pre-cardioversion and then following them post-cardioversion. We can see that the information was obtained using trans-telephonic monitoring of the electrocardiogram. All that mattered was to suppress the arrhythmia. No other significant questions were asked. We did find out, of course, that cardioversion was improved, that spontaneous or drug-induced cardioversion occurred better with these drugs, and we particularly found out that amiodarone beat its competitor sotalol, and both were better than placebo. But it took a little while for us to appreciate that simply trying to record intermittent arrhythmia was a problem. We needed to know more about the density of the arrhythmia. How often did it occur? How much of the time did patients spend in atrial fibrillation? And therefore, we needed to take opportunity of patients who were fitted with implanted devices which could log the prevalence of the arrhythmia. So an implantable cardioverter-defibrillator fitted in the SCUD-HEF trial, a trial of patients with New York Heart Association class 2-3, left ventricular ejection fraction 35% or less, offered that opportunity. Patients were randomized to an ICD, amiodarone, or placebo. During the trial, the 2,300 patients who started off in sinus rhythm, 11% of those developed atrial fibrillation. And the development was different in these groups. We saw that the patients who had the ICD had the highest rate of development. Those who were treated with amiodarone had the lowest rate, and the placebo was in between. The aggravation of atrial fibrillation is caused by the pacing interventions and the shock interventions, and amiodarone is presumably responsible for the reduction of atrial fibrillation in this group of patients. We also, of course, found out in the same trial, one of the primary purposes of the trial, that amiodarone was not particularly good at reducing all-cause mortality because it shared this same rate with placebo. And what was more, we discovered that although there was a marginal gain, not statistically significant, in New York Heart Association Class 2 patients, this did not extend to New York Heart Association Class 3 patients. So here we were using the device interventions as the outcome event rather than the log of the device that we had used for the primary purpose in the AF study. So amiodarone became by far the most popular antiarrhythmic drug. And this is actually a picture hanging in MoMA, the Museum of Modern Art in New York. It's painted by Damien Hirst. And he wrote, "I can't understand why some people believe completely in medicine and not in art," without questioning either. He provided a suite of paintings which he called The Last Supper, referring to the way in which medicinal drugs are becoming a regular part of everyday life, as common as the food. And he chose from the cardiovascular drug panoply amiodarone, but called it corned beef. Now, the picture that I took in the gallery was on a poor camera, and it was pretty awful, so I recreated it. So this isn't the original. It certainly won't sell as much as Damien Hirst's painting, but it's pretty accurate. And it's just a signal of the fact that amiodarone became an important drug in the cardiovascular arena. Now, moving onward into the arena in which atrial fibrillation became the main target of therapy, we can see that there was a distinct movement towards doing more than cosmetically obliterating atrial fibrillation. It became important to demonstrate that that was associated with an improvement in important clinical outcomes. The Athena trial looked at a new drug known as dronedarone in order to investigate that possibility. Using a primary endpoint of cardiovascular hospitalization and death, patients with at-risk atrial fibrillation were randomized to placebo or dronedarone and followed. You can see there was a reduction in this primary endpoint of 24%, which was highly clinically significant. This is the first and only trial in which such an outcome has been demonstrated in atrial fibrillation with any drug. So it was a landmark, and a landmark which is now set as an important concept in the drug development of any agent for the management of atrial fibrillation. A bitter pill for the pharma industry to swallow because this is an expensive approach. But amongst these patients, there were those who remained, despite the antiarrhythmic, in atrial fibrillation throughout the whole trial. And they're described as permanent atrial fibrillation, in quotes. And we can see, if we follow those patients, that there is a similar relative risk reduction, in this case 26%, again statistically significant, but you can see that we're no longer dealing with a properly randomized group of patients. There are far more patients, for example, in the placebo than in the dronedarone wing. So this, of course, was not a significant result. It wasn't something that we could rely on. It was only hypothesis generating. And the hypothesis was that even in patients with atrial fibrillation, dronedarone might have other advantageous effects, perhaps suppressing ventricular arrhythmia, for example, reducing hypertension and reducing the heart rate in atrial fibrillation, etc. And so it was decided to run another trial recruiting patients with permanent atrial fibrillation, and that was known as the Palace trial. It was intended to randomize some 11,000 patients with permanent atrial fibrillation and more than 6 months duration, and with no unstable heart failure. But when this randomization occurred, it soon became evident that there was increased hazard associated with both of these co-primary endpoints in the dronedarone treated group. Here we can see 43 to 19 events, 127 to 67 events. So the hypothesis didn't pan out here either. But subsequent analysis demonstrated it was not possibly not entirely due to dronedarone, because if we look at the outcome event of all-cause mortality and cardiovascular death and arrhythmic death, we can see that it is the increased hazard, which was 12 events with regard to mortality, was seen only in patients who were co-administered digoxin. And that applied to cardiovascular death and applied to arrhythmic death, where the difference was 11 instances to 0. And therefore, some drug-drug interaction is possibly relevant because dronedarone, a PGP inhibitor, for example, will increase the plasma concentration of digoxin, of dronedarone. So let's move now to another approach that has occurred in the anti-arrhythmic arena, a curious development. An anti-anginal drug known as ranolazine, which is a late sodium current inhibitor, was found by chance because of Holter monitoring and clinical events in a large clinical trial to suppress atrial fibrillation. It was a post hoc analysis, but it did. Now, this late sodium current inhibitor, it affects this part of the sodium current, not the peak current, which is what flecainide, for example, affects, but this late part is responsible for at least 50% of the sodium ingress into the cell during the action potential. And you can see the late sodium current blocker ranolazine has its effect predominantly on the late sodium current as opposed to the peak sodium current. In atrial fibrillation, the late sodium current is increased and now is responsible for bringing into the cell about 3 or 4 or 5 times as much sodium as the peak sodium current. You can see also that with ranolazine, this can be reversed. So ranolazine will take back this late sodium current to the pre-treatment level. Now, if we see that this happens, of course, it's likely that after depolarizations, whether they're early or delayed, will also be suppressed. And this is one of the mechanisms that will produce the triggers, at least, for atrial fibrillation. So there's a good basis for the fact that this anti-anginal drug might also be anti-arrhythmic. So it was no surprise that this became used by physicians. It was investigated by groups of physicians, but no proper study was ever done by the pharma company. Another limit, in the sense that this drug was then the property of Gilead, a company with no cardiovascular experience, and a company that was in it for a very quick buck, so to speak. So they never investigated the drug, except they were persuaded to do a study known as Harmony, which was a combination of dronedarone, of which we've just spoken, with ranolazine. And again, using a pacemaker to measure the burden of atrial fibrillation. And we can see that in this study, that the drug dronedarone or ranolazine alone has only a relatively minor effect on the change of the atrial fibrillation burden, but the combination, particularly 750 of ranolazine and 225 of dronedarone, was highly effective, reducing the burden by some 60%. And this was clearly more than a simple arithmetic sum of the two effects, probably related to the conversion of each of these drugs from relatively single-channel drug effects into multi-channel drug effects. But this, again, is the best example of a drug combination study in the anti-arrhythmic world. Curiously, such studies have never been done properly, and yet physicians are often inclined to add one anti-arrhythmic agent to another, a highly dangerous way to practice, but one that is often done in the real world. And the reason, of course, brings us back to the starting point, because these drugs are essentially not only anti-arrhythmic, but potentially pro-arrhythmic. If we look at this Cochrane analysis of drugs used for atrial fibrillation and published by Lafuente, we can see that pro-arrhythmia occurs in all of these anti-arrhythmic drugs, in many cases highly significant results. And we can see also that overall mortality is increased with anti-arrhythmic drugs, not reduced. There might be one example, dronedarone, which has been associated with lower mortalities, and yet we just discussed the higher risk of taking dronedarone in the permanent atrial fibrillation population. But notice that quinidine is associated with a statistically significant increase of about 3-fold in mortality, and so too is sotalol, a drug which many of you may also have had your hands on. You may also have used sotalol, but I encourage you not to do so. So let us now take a look back some 10 years. I first gave a lecture at the limits about, I think, 8 years ago, and this slide comes from 2 years before that, and I showed it at that meeting. This is what I described as a smorgasbord of anti-arrhythmic agents. The ones at the top you will know well. You may not know most of these on the left-hand side, and you certainly won't know most of these because I've not even put names to these drugs, but they were all new targets for the management of atrial fibrillation. If we now take a look at what is left, and to clarify it, let me show the next slide. You can see that the world of antiarrhythmic drug development has been a huge disappointment. These agents are decades old. In the 20th century, dofetilide, dronedarone, vernicalin, and the late surgeon blocker, ranolazine, is all we have to show for it. But help is at hand. We have yet new anti-arrhythmic agents. I don't want to spend a lot of time on these agents, but I will just show you the list of them. We have a synthetic omega-3 fatty acid in phase 2-3. We have a small conductance calcium-activated potassium channel inhibitor, a new target for atrial fibrillation, also in phase 2, phase 3. We have another multi-channel blocker, very similar to amiodarone, but without the iodine, known as HBI 3000. We have intranasal etripamil, a calcium antagonist, L-type calcium antagonist. We have inhaled flecainide. And we have Wenshin Kali, another highly limiting problem. How do you develop a Chinese herb? Although it's effective, can it ever be developed as a pharmaceutical? It's unclear what component of that herb, and there are many thousands of components, is responsible for the antiarrhythmic effect. But importantly, it's very noticeable that none of these new antiarrhythmic drugs is owned by big pharma. All of these molecules are in the hands of small biopharma companies. And let's have a look at a couple of them just for fun. This is the so-called puff in the pocket, inhaled flecainide. Now, it's meant to be something that you can take with you, and when you develop atrial fibrillation, you can just inhale some flecainide, you get it right to the site of action, and the arrhythmia will stop within 30 seconds. That may be true, but this is hardly something that you can put in your back pocket. It might fit in a briefcase or a large handbag, but not something that would go in the pocket. But it does work, at least in animals, and there are now good human studies, not reported, but good human studies demonstrating similar efficacy. You can see that with inhaled flecainide, the arrhythmia will terminate and normal sinus rhythm will resume. But this slide gives me considerable worry. It's a slide from a company. And we can see, if we look at it, that this part and this part are identical, this part and this part are identical, and somehow this is normal sinus rhythm, this is atrial fibrillation. So I think the slide is not telling us the entire truth, but nonetheless, I have it from the investigators currently working with this drug in humans that it is effective when administered in this way. And this is etripamil. Here, paroxysmal supraventricular tachycardia is happening. Suddenly your heart races, bang, 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 bang. You simply sniff now, not puff, but you sniff etripamil, and within, as you see, a couple of minutes, very fast conversion of paroxysmal supraventricular tachycardia. This drug is also now in phase 2, phase 3 studies and is very promising. Here's Wenshin Kali that I mentioned to you. Some basic science studies abound with this drug. There are some clinical studies done coming from China, but so far nobody in the Western world has decided to further investigate this compound. Now, of course, there are many other approaches to anti-arrhythmic management. One interesting concept is the concept that anticoagulation, particularly with factor Xa inhibitors and with direct thrombin inhibitors, will prevent the fibroblast formation that occurs because of the hypercoagulable inflammatory state in atrial fibrillation. There's a large registry which is currently investigating this, known as RACE5, and there are two studies, not primarily intended for this purpose, known as NOAA and Artesia, which are looking at factor Xa inhibitors in patients with small spells of atrial fibrillation to see how they progress. And we will have some good information on whether anticoagulation as such can reduce the likelihood of atrial fibrillation and its further progression. We're also interested in some sub-study information that comes, for example, from the BEST study. And you remember the BEST study was in patients with heart failure involving bucindolol, which of course is a beta-1 adrenergic blocker, which also reduces the number of beta-1 receptors and norepinephrine concentrations. In this sub-study, we can see that this drug certainly reduces the likelihood of atrial fibrillation developing, as you can see on the left-hand side, and that this effect is almost entirely due or seen in the homozygous beta-1 adrenergic receptor 389 arginine rather than the heterozygous arginine glycine, where there is no effect noted. Now, this is a remarkable finding. Again, it's entirely post hoc, but there is now a large trial going ahead to investigate this further. Now, let me turn for a last swing at the antiarrhythmic arena to botulinum toxin, not now for wrinkles on the face, but wrinkles in the heart, particularly in the stellate ganglia of the heart. Botulinum toxin, as you know, blocks the release of acetylcholine. It can be given to the stellate ganglia. There are 4 or 5 of these ganglia around the atria in the heart. They can be injected at the time of surgery so far, and when you do that, you can see that even long-term studies show a remarkable reduction in the likelihood of the appearance of atrial fibrillation, placebo, botulinum toxin, placebo, botulinum toxin, 36 months. Just a few words about the potential genetic element, because of course we all expect that a more purposeful and precise approach to the management of cardiac arrhythmia would be much better. Here's a good example, one of the very few in cardiology. This is long QT3 syndrome. This is due to an upregulation of the late sodium current in the sodium channel. And that, if you put more and more sodium into the channel, will prolong the QT interval. And if you give a drug like ranolazine to prevent the late sodium current, you will see that you shorten the QT interval. This is being given to, in this particular example, about 5 patients who had long QT syndrome, but it's now used clinically because it shortens the QT and suppresses Torsades de Pointes, a highly precise approach to the management of this single genetic disorder. We, of course, have other approaches where we can use genetic markers to tell us whether a drug may be more effective. In this particular example, we're looking at mRNA in the left atrium, and we can find that some patients have very low levels of mRNA in the left atrium. All of these patients are coming for ablation of atrial fibrillation, and the patients who have low levels of mRNA have got relatively depolarized resting membrane potentials, and if you give flecainide to these patients, you extend the post-repolarization refractoriness far more than in other patients, a good signal by which to choose patients for whom flecainide may be valuable. And of course, we're all waiting for much more targeted genetic approaches to therapy. As always, the problem is the vector, but cardiologists are good at getting at the left atrium. One way or another, they can certainly get genetic modifications to the left atrium, and of course, there are many potential targets. Time is too short for me to spell them out. So let's move now into the device arena for a short while. And I think that I'd like to show you first the issue of ablation for atrial fibrillation. Now, this is a highly popular technique. It is pretty effective, but does it actually reduce important cardiovascular outcomes? It can, admittedly, reduce symptoms, reduce the likelihood of atrial fibrillation, but does it do anything to stop people dying? Well, here's a result of a trial known as CASTLE-AF, recruiting 3,000 patients, not recruiting, but looking at 3,000 patients and enrolling nearly 400 and randomizing them to ablation or conventional therapy. All the patients had ICDs to log atrial fibrillation. They also had low ejection fraction New York Heart Association Class 2. You can see that they were followed for 60 months. A lot of patients crossed over on both sides, but the results were, as you see, highly in favor of the group randomized to ablation therapy rather than conventional therapy, with a 38% relative risk reduction in the primary endpoint, which was a composite of hospitalization and mortality. And if we look at all-cause mortality alone, a 47% reduction. Unfortunately, the trial is questioned because of all of these crossovers and the difficulty with recruiting patients and so on and so forth, but it is at least a signal that we may be able to help with ablation. Very recently, we've had the publication of the Cabana trial, a similar trial undertaken in about 2,000 patients, randomized to ablation or to drug therapy. These are all comers, not just those with heart failure. And we can see that if we look at the primary endpoint, again, a composite, another limitation in the antiarrhythmic arena, that composites are the only way that you can accumulate sufficient events. You can see that ablation was not superior to drug therapy, and that drug therapy could be antiarrhythmic or could be rate control. And even if we use a per-protocol analysis, again, we do not see a strong effect, but it is just statistically significant. If, however, we take yet another step, and this is the step advocated by C. Everett Koop, the former US Surgeon General, who said if patients don't take drugs, they don't work. So the people doing the Cabana trial said that we must look at only the patients who had ablation and compare them with the patients that had drugs. Of course, this leaves us again with non-randomized data. But if we do so, we can see that those who had an ablation did remarkably well compared with those that did not. In my view, the trial is a failed trial. Again, it's another signal that ablation may be highly favorable, but not, in my view, conclusive. But those who want to ablate will certainly hang on to this data very fiercely. So let me then move to a few technical advances, because this is really part of the arrhythmia scene. I'd like to show you, for example, how mapping of the heart in a patient with atrial fibrillation has become a highly sophisticated business. It usually comes as a black box, or if not a black box, a highly complicated mathematical equation, which neither you nor I understand. But nonetheless, you can see that the pictures are very pretty, in this example showing a rotor, and the implication is if you ablate this spot for the rotor, then the atrial fibrillation will no longer be possible. Let's have a look at another technique used for ablation. Instead of just radiofrequency or cryoenergy, you can use laser, and you can use many other forms, but here, electroporation is the technique which is being used. This is a DC current applied frequently at relatively low voltage. It disrupts the bilipid membrane layer of cardiac cells and allows them to become depolarized, and permanently so if you do it enough. And this particular technique is to put onto a glove a series of electrodes, or put onto a fingertip a series of electrodes and move them around the back of the heart, in this case ablating not the myocardium but the stellate ganglia, which we've referred earlier. Alternatively, we might go to some really vicious cryotherapy. Here's an example of liquid nitrogen, which is critical, meaning that it goes down to minus 196 degrees centigrade. This is highly effective. You can see that they're using catheters with multipoles, up to 20 electrodes, and they use stylets which are bent in various ways so that they can be moved around in the left atrium, and you can put it almost anywhere. You do have to put a warm balloon into the esophagus to stop the esophagus cooling down, freezing, and left atrial esophageal fistulae developing. But provided that you take care, it's said to be excellent from a safety and an efficacy perspective. Alternatively, we can use stereotactic radio ablation. Here we're just looking at fairly conventional irradiation, but you can use 12 carbon particles as well. You see the technique that you aim from all over the surface of the body at one spot in the heart, and you can destroy that spot, in this case the AV node. You see the development of AV block, and here's the target. Of course, there are some advantages and disadvantages, but the main disadvantage, of course, is that with radiation therapy, it may take days to months to achieve the full effect of the therapy. And this is a somewhat dangerous approach at the moment and has not yet got much into the clinical arena. Now, turning to ICDs just for a moment, the Implantable Cardioverter-Defibrillator was shown to be clinically effective some 20 or 30 years ago. You'll remember the MADE-IT trial, for example, which showed in ischemic heart disease that patients with a low ejection fraction could live much longer if they were treated with an implantable cardioverter-defibrillator. The same became shown at a somewhat later date, and not as convincingly, with non-ischemic heart failure. And the Danish trial, which was released just a couple of years ago, demonstrated that in patients with non-ischemic cardiomyopathy and ejection fraction 35% or less, that there was no difference, no statistically significant difference in death rates. There was a small difference, a large relative risk reduction, but a small absolute risk reduction in cardiovascular mortality, which was sudden in nature. There were exceptions to this. The younger patients did have a reduction in all-cause mortality, but it put a big question mark over the trials which in the past demonstrated efficacy. This is a limitation. How far can we think of those trials as being relevant today? We've had major changes, as you well know. We can select patients in much better ways. For example, just looking at late gadolinium enhancement will allow us to find patients with fourfold risks of ventricular arrhythmia and sudden death on top of their ejection fraction risk. So we can certainly do other ways of finding patients who we might fit an ICD to. Now, here are two exemplary trials of the ICD which are happening today. On the left-hand side is RESET SCD. And it's been undertaken because since MADIT, for example, we have ARNIs, we have MRA, which reduce all-cause mortality and sudden cardiac death. We have statins and beta blockers. We have much better management of coronary artery disease. So if we take patients that fit the original MADIT criteria, randomize them today with optimal medical therapy, will we have the same outcome? On the other hand, is there a better way of identifying patients? We might not rely on left ventricular ejection fraction, but instead take some autonomic measures, in this particular instance, in the REFINE ICD trial. It's interesting to see how these trials are funded. This trial, which questions the established indications for the ICD, is not funded by industry. You wouldn't expect it. It's funded instead by healthcare providers who can actually save money from not implanting ICDs. This trial, on the other hand, which is seeking to establish new indication, is readily funded by industry, and that's no surprise, I think, to anyone. Let me just say a word about pacemakers. As you know, originally we had pacemakers fitted in the subpectoral position. We're now putting them into the apex of the right ventricle, for example. Alternatively, we can fit them against the His bundle and pace the His bundle. We can put them in the left ventricle. We can combine these devices, one in the atrium, one in the ventricle. Could be right atrium or left atrium, could be right ventricle or left ventricle. It could be left ventricle and right ventricle to achieve cardiac resynchronization therapy. We could also use other leadless pacing devices. This, for example, is an acoustically coupled electrode system. This implanted onto the endocardial surface of the left ventricle, this under the skin, powered by this battery. Acoustic coupling powers this amplifier and stimulator and allows pacing. All of these are recent developments, they're all going ahead, and also we're seeing, for example, the combination of the subcutaneous defibrillator, no lead in the heart, and implantable pacemakers, which can be ganged together and which will communicate with each other by pulses given during the ventricular refractory period, allowing one implantable device to talk to another implantable device. The world of arrhythmias is very exciting. It's had lots of disappointments. It will have more. But to return to the point that you made, Peter, at the start of this talk, we've just had the results of the Apple Heart study. Some 419,000 participants in this trial, recruited in a short period of time, followed wearing the Apple Watch, the original version just recording the so-called photoplethysmographic signal, the modern version recording an electrocardiogram. But the observation found some 2,100 patients who had pulse irregularities, a significant proportion of which had atrial fibrillation. Others had ventricular premature contractions, and yet others had no real abnormality. But this is the kind of information you can now get from the Apple Watch, a complete printout of the arrhythmia, in this case, atrial fibrillation. So my warning is to you, although you may have no interest in cardiac arrhythmia, it will come to your door. Thank you very much for your attention.